There are two pathways that affect Alzheimer's disease, Wnt signaling and PPAR. This video focuses on the Wnt signaling pathway. Wnt stands for Wingless Related Integration Site. The pathway Wnt signaling is crucial to helping cells survive, embryonic development, and synaptic activity. Wnt is an important pathway at the synapse. Synaptic activity in the Wnt pathway sends electrical and chemical signals that are required for learning and memory. It is needed for synaptic plasticity and maintaining the adult brain. Wnt signals are usually active in early development and then later in life for tissue development. The Wnt pathway conducts paracrine signaling, aka signaling nearby cells. Evidence suggests Wnt signals are stem cells with multiplying and self-renewing features. Wnt ligands bind to fizzled and LRP5 and LRP6 receptors. Wnt signaling pathway protects against destruction of neurons made by amyloid beta proteins. The pathway stops the destruction of beta-catenin. Beta-catenin regulates gene transcription.
There are no gene mutations present in the correct signaling mechanism. There are also no chemical or electrical imbalances in the signaling pathways. Wnt binds to transmembrane protein receptors on the surface of the cell, which activate other signaling pathways. These pathways regulate gene transcription and the release of calcium +2 ions. There is a sufficient amount of LRP6, which regulates production of amyloid beta and communication between neurons. There is no hyperphosphorylation of tau, and the B-catenin protein sends signals to the nucleus that lead to the transcription of Wnt genes, which regulate cell life. Amyloid beta peptide is not produced, so there is no buildup of amyloid plaques. The amyloid precursor protein is an integral protein that makes SAPP, which leads to normal synaptic signaling, synaptic plasticity, learning, memory, and emotions. When it is diseased, it leads to plaque formation. At the synapse, the neurons generate an action potential, causing a neurotransmitter to be released. Receptors on the dendrite bind with the neurotransmitter. The neurotransmitters are released and go back up into the first neuron in a process called reuptake. The axon carries the signal to other neurons, and the process starts again.
Signaling is known to have a key part in the survival of cells, the development of embryos, and the activity of synapses. These signals are vital for learning and memory. Studies have shown that Wnt signaling is suppressed in Alzheimer's. Wnt signaling suppression results from a prime defect, a deficiency in low-density LPR6, a protein which manages amyloid beta production and neuron communication. Therefore, an LRP6 deficiency results in a toxic accumulation of amyloid and hinders neurons' ability to communicate. Synaptic failure occurs toward the beginning of Alzheimer's disease. It is proposed that soluble AB oligomers are at fault for the synaptic pathology that takes place before plaque buildup and the death of neurons. Alzheimer's is identified by extracellular amyloid deposits, intracellular neurofibrillary tangles, cholinergic deficits, synaptic loss, inflammation, and extensive oxidative stress. Neurofibrillary tangles and amyloid plaques lead to the death of neurons and a decreased number of synaptic proteins, which results in memory loss and changes in cognition. Thanks for watching!